The relentless march of time brings with it a profound biological transformation, particularly within the invisible fortress of our immune system, a phenomenon that becomes acutely apparent as we cross the threshold of 65 years of age. For decades, the scientific community and the general public alike have accepted this gradual decline in vitality and susceptibility to illness as an inevitable consequence of aging, a mandatory surrender to the biological clock. However, a revolutionary paradigm shift is currently unfolding in the realm of geriatric immunology and nutritional biochemistry, fundamentally challenging this archaic assumption. At the very core of this scientific renaissance is the strategic, highly selective supplementation of a singular, master regulatory trace mineral, zinc. By applying a targeted, selectively bound zinc protocol, we are now discovering that it is entirely possible to combat, and in many ways reverse, age-related immunosenescence, thereby restoring vital T-cell function and rejuvenating thymic output in the aging body. This is not merely a theoretical postulation but a highly intricate, biomechanically validated intervention that we continuously visualize and validate through advanced 3D cellular animations, allowing us to witness exactly how specific mineral binding allows zinc to bypass the hostile environment of the aging digestive tract, cross the formidable intestinal barrier, extinguish the smoldering fires of chronic systemic inflammation, and ultimately rebuild the biological defense systems that are absolutely essential for maintaining long-term physiological independence in our golden years. To truly comprehend the magnitude of this immunological resurrection, we must first understand the devastating reality of what happens to our internal defense forces as we age, a process scientifically termed immunosenescence. As we navigate past our sixth decade of life, the immune system undergoes a radical and detrimental restructuring. The most critical casualty of this process is the thymus gland, a small, bilobed organ situated just beneath the sternum that serves as the master university for our adaptive immune system. In our youth, the thymus is a bustling hub of cellular education, taking immature white blood cells produced in the bone marrow and rigorously training them to become naive T cells, the elite special forces of our immune system that are capable of recognizing and destroying novel, never-before-seen pathogens. However, beginning as early as puberty and accelerating dramatically by age 65, the thymus undergoes a process known as thymic involution, whereby its active, T-cell-producing lymphatic tissue is progressively replaced by inert adipose, or fat, tissue. In the high-definition 3D cellular animations we use to illustrate this process, the visual contrast is stark. A youthful, robust thymus appears as a vibrant, highly vascularized network teeming with cellular activity, whereas the aged thymus resembles a dormant, fatty remnant struggling to produce even a fraction of its former cellular output. This dramatic collapse in thymic output leads to a perilous depletion of naive T cells in the peripheral bloodstream. Consequently, the aging immune system becomes increasingly reliant on a stagnant pool of memory T cells, which have become exhausted and hyperspecialized from a lifetime of fighting past infections, leaving the elderly body dangerously defenseless against new viral strains, bacterial mutations, and the spontaneous emergence of malignant cancer cells. Compounding this catastrophic loss of adaptive immunity is a parallel, equally destructive phenomenon known as inflammaging. Inflammaging is a state of chronic, low-grade, sterile inflammation that silently ravages the tissues of the elderly body, driven by an overactive innate immune system that constantly churns out destructive pro-inflammatory cytokines such as interleukin-6, tumor necrosis factor alpha, and C-reactive protein. This constant state of systemic alert not only damages healthy tissues and accelerates cardiovascular and neurodegenerative diseases but also acts as a biological static, blinding the remaining immune cells to actual threats. The aging individual is thus trapped in a paradoxical state. Their immune system is simultaneously hyperactive, destroying their own tissues through inflammation, yet profoundly immunodeficient, unable to mount an effective, targeted response against genuine infectious invaders. It is within this bleak landscape of cellular exhaustion and inflammatory chaos that the true miracle of selective zinc supplementation emerges as a beacon of biological restoration. Zinc is not merely a static building block, it is a dynamic, intracellular signaling molecule, a crucial catalytic cofactor for over 300 distinct enzymatic reactions, and the ultimate master conductor of the immunological orchestra. However, simply consuming generic, commercially ubiquitous forms of zinc, such as zinc oxide or poorly formulated zinc sulfates, is a futile endeavor for the aging body. The gastrointestinal tract of an individual over 65 undergoes significant physiological changes, including a marked reduction in gastric acid secretion, known as hypochlorhydria, and a thinning of the mucosal lining, which drastically impairs the body's ability to cleave, absorb, and utilize standard mineral supplements. Furthermore, the modern diet is replete with phytates, oxalates, and tannins found in grains, legumes, and certain vegetables, which act as anti-nutrients, relentlessly binding to free zinc in the digestive tract and forming insoluble complexes that are unceremoniously excreted from the body, leaving the individual deeply deficient despite adequate dietary intake. To circumvent this profound biological roadblock, we must turn to the science of selective mineral binding, a method that employs targeted, organically chelated forms of zinc, such as zinc picolinate, zinc bisglycinate, or highly specialized zinc L-carnosine complexes. By utilizing advanced 3D cellular animation, we can visualize the extraordinary journey of these selectively bound zinc molecules as they are ingested and travel down the esophagus into the hostile, low-acid environment of the elderly stomach. Unlike raw, unchelated zinc, which would be instantly neutralized or bound by dietary anti-nutrients, the selectively bound zinc is shielded by its attached amino acid or organic acid chaperone. This molecular armor protects the zinc ion as it transitions into the small intestine, specifically the duodenum and jejunum, where the intricate dance of absorption must occur. Here, the animation zooms in on the brush border of the intestinal lumen, revealing the microscopic, finger-like projections known as villi, and the even smaller microvilli, which are lined with specialized absorptive cells called enterocytes. The selectively bound zinc complex approaches the apical membrane of the enterocyte, seamlessly evading the predatory grasp of phytates. Because the zinc is chelated to a molecule that the body readily recognizes and desires, such as the amino acid glycine in zinc bisglycinate, it effectively tricks the enterocyte into actively transporting the entire complex across the cellular membrane, utilizing efficient amino acid transport channels rather than relying solely on the easily saturated and often genetically downregulated ZIP4 zinc transporter proteins. Once safely inside the protective cytoplasm of the enterocyte, the organic chaperone is gently cleaved away, releasing the elemental zinc ion. However, the journey is far from over, as free zinc is highly reactive and must be carefully managed to prevent cellular toxicity. The enterocyte rapidly synthesizes a specialized, cysteine-rich binding protein called metallothionein, which acts as an intracellular taxicab, safely sequestering the zinc ion and ushering it toward the basolateral membrane of the cell. From here, specialized efflux proteins known as ZNT1 actively pump the zinc out of the enterocyte and into the hepatic portal vein. As the zinc enters the systemic circulation, it is immediately bound by serum albumin and alpha-2 macroglobulin, which serve as heavy-duty transport vehicles, navigating the newly absorbed zinc through the labyrinthine network of the cardiovascular system, delivering this vital payload to the tissues that are most desperately starved for its regenerative power. The primary destination for this life-giving mineral is the atrophied, aging thymus gland. As the animated sequence transports us inside the thymic microenvironment, we witness the profound, almost miraculous impact of the newly delivered zinc. Zinc is the absolute, non-negotiable prerequisite for the biological activation of a critical thymic hormone known as thymulin. In the zinc-deficient elderly body, the thymus may still produce the base peptide of thymulin, but without a zinc ion firmly bound to its molecular structure, the hormone remains biologically inert, utterly incapable of stimulating T-cell maturation. However, as the selective zinc protocol floods the thymic tissue with highly bioavailable zinc ions, these ions perfectly slot into the molecular structure of the inactive thymulin peptides, instantaneously transforming them into fully active, potent hormones. This surge in active thymulin sends a powerful, cascading biochemical signal throughout the thymic tissue, effectively waking the organ from its senescent slumber. The active thymulin acts upon the local thymic epithelial cells, stimulating them to resume the critical work of thymopoiesis, the generation of new T cells. We can visually track this process as precursor cells arriving from the bone marrow are suddenly enveloped in a nurturing, hormone-rich environment. They begin to rapidly divide and differentiate, undergoing rigorous positive and negative selection processes to ensure they can recognize foreign antigens without attacking the body's own tissues. This zinc-induced thymic rejuvenation culminates in a steady, revitalized output of brand new, highly adaptable naive CD4+ helper T cells and CD8+ cytotoxic T cells, which are subsequently released into the peripheral bloodstream. This fresh influx of naive T cells dramatically shifts the immunological repertoire of the aging individual, diluting the stagnant pool of exhausted memory cells and restoring the body's capacity to mount a vigorous, highly specific defense against novel respiratory viruses, bacterial pneumonias, and newly emerging pathogenic threats that so often prove fatal to the elderly. Simultaneously, the Selective Zinc protocol initiates a second, equally critical campaign across the entire biological landscape, the aggressive suppression and reversal of chronic inflammaging. As the bioavailable zinc penetrates the membranes of circulating innate immune cells, such as macrophages and dendritic cells, it exerts a profound, epigenetic regulatory effect. Within the cytoplasm of these cells, zinc acts as a direct inhibitor of a master transcription factor known as nuclear factor kappa B, or NFKB. In the aging, inflamed body, NFKB is constantly active, translocating into the cell nucleus and relentlessly switching on the genes responsible for producing the destructive inflammatory cytokines that fuel inflammaging. However, as intracellular zinc concentrations rise, zinc firmly blocks the activation pathways of NFKB, essentially serving as a master biochemical off-switch for systemic inflammation. Our 3D cellular models beautifully illustrate this molecular intervention. We see the fiery red, hyperactive macrophages, furiously pumping out toxic levels of interleukin-6 and tumor necrosis factor alpha, suddenly calm down as zinc ions flood their interior. The NFKB molecules are halted in their tracks, prevented from entering the nucleus, and the production of inflammatory cytokines immediately plummets. This dampening of the inflammatory fire has cascading, systemic benefits that extend far beyond simple immune function. As the burden of chronic inflammation is lifted from the body, the structural integrity of the vascular endothelium improves, reducing the risk of atherosclerosis and cardiovascular events. The blood-brain barrier tightens, protecting the fragile neural tissues from inflammatory damage and preserving cognitive function, and the constant, debilitating drain on the body's metabolic energy reserves is finally alleviated, resulting in a profound, tangible surge in physical vitality and stamina for the elderly individual. The success of this selective zinc supplementation protocol, however, does not exist in a vacuum. It requires a holistic, deeply integrated approach that considers the intricate interplay between nutrition, lifestyle, and the unique psychology of aging. To maximize the intracellular delivery and efficacy of zinc, the protocol must be synergistically paired with specific nutritional cofactors. One such vital cofactor is quercetin, a naturally occurring dietary flavonoid found in capers, red onions, and apples. Quercetin acts as a powerful zinc ionophore, a specialized molecule that embeds itself into the lipid bilayer of the cellular membrane and creates a temporary, highly conductive channel, actively facilitating the rapid transport of zinc ions from the extracellular fluid directly into the cytoplasm of the target cells. This synergy is particularly crucial for equipping cells to rapidly shut down viral replication, as high intracellular zinc concentrations have been definitively shown to inhibit RNA-dependent RNA polymerase, the very engine that respiratory viruses use to hijack our cells and multiply. Furthermore, careful attention must be paid to the delicate balance between zinc and copper, as long-term, high-dose zinc supplementation can competitively inhibit copper absorption in the gut, leading to a secondary copper deficiency that can manifest as anemia and neurological impairment. A scientifically sound selective zinc protocol for the elderly meticulously accounts for this biological seesaw, ensuring that supplemental zinc is either carefully cycled or appropriately paired with microdoses of organically bound copper to maintain perfect mineralogical homeostasis. Beyond the realm of nutritional biochemistry, the lifestyle habits of the elderly individual play an enormous role in optimizing the restorative power of this protocol. The circadian rhythm, the body's internal 24-hour clock, is deeply intertwined with immune function and zinc metabolism. Aging is frequently accompanied by severe disruptions in sleep architecture, characterized by fragmented sleep, reduced deep restorative slow-wave sleep and diminished melatonin production. Melatonin is not merely a sleep hormone, it is a potent antioxidant and an immunomodulator that works in direct synergy with zinc to repair cellular damage incurred during waking hours. Therefore, optimizing sleep hygiene, through strategies such as maximizing early morning sunlight exposure to regulate the suprachiasmatic nucleus, minimizing artificial blue light in the evening, and establishing strict, consistent sleep schedules, is absolutely vital to ensure that the body is in a correct parasympathetic, restorative state to utilize the zinc provided by the protocol. Furthermore, integrating regular, appropriately scaled physical exercise is a potent force multiplier for immune rejuvenation. Exercise, particularly moderate-intensity resistance training and aerobic activity, induces transient, beneficial microstresses on the body that stimulate the mobilization of immune cells from marginalized pools in the lungs, spleen, and bone marrow into the central circulation. This increased hemodynamic shear stress physically sweeps the newly minted, zinc-activated naive T cells throughout the vascular network, ensuring they continuously patrol every corner of the body. Additionally, contracting skeletal muscle tissue releases highly beneficial signaling proteins known as myokines, which actively counteract the destructive inflammatory cytokines associated with inflammaging, further amplifying the anti-inflammatory effects of the zinc protocol. We must also profoundly address the psychological dimension of aging and immune decline, a factor that is too often ignored by purely mechanistic medical paradigms. The psychological burden of advancing age is frequently characterized by a pervasive, creeping anxiety regarding one's physical vulnerability. The fear of contracting a severe respiratory infection, the dread of prolonged hospitalization, and the ultimate terror of losing one's physical independence and becoming a burden to loved ones can create a chronic, unyielding state of psychological stress. This chronic emotional stress activates the hypothalamic pituitary adrenal axis, resulting in a continuous, low-grade secretion of cortisol. While cortisol is necessary in acute, fight-or-flight situations, chronically elevated cortisol is fiercely immunosuppressive, literally commanding the thymus to accelerate its involution and signaling the bone marrow to halt the production of new immune cells. Furthermore, high psychological stress severely alters gastrointestinal motility and reduces the secretion of digestive enzymes, which completely undermines the careful work of selective mineral absorption, preventing the targeted zinc from ever reaching the bloodstream. Therefore, the implementation of a selective zinc protocol must be accompanied by psychological empowerment and stress mitigation strategies. By educating the elderly individual about the precise, scientifically validated mechanisms through which they are repairing their immune system, we transform them from passive victims of the aging process into active, empowered participants in their own biological restoration. When an older adult views our 3D animations and visualizes the zinc actively repairing their thymus, suppressing cellular inflammation, and building a new army of defensive T cells, it generates a profound psychological shift. This visualization fosters a powerful sense of self-efficacy, reducing the existential anxiety associated with aging and effectively lowering the chronic cortisol burden. The brain and the immune system are engaged in constant, bidirectional communication. An empowered, hopeful mind creates a neurochemical environment that physically enhances the efficacy of the immune response. Furthermore, zinc itself plays a direct, deeply structural role in cognitive and psychological well-being. The hippocampus, the region of the brain responsible for memory formation, emotional regulation, and spatial navigation, contains the highest concentration of zinc in the entire central nervous system. In this neural epicenter, zinc is sequestered in synaptic vesicles and released alongside the neurotransmitter glutamate, where it meticulously regulates synaptic plasticity, the physical mechanism underlying learning and memory. By rectifying age-related zinc deficiency through our selective protocol, we not only rebuild the immune system but also directly nourish the neural architecture of the aging brain, frequently resulting in reported improvements in mental clarity, mood stabilization, and a reduction in the depressive symptoms that so commonly afflict isolated elderly populations. The profound, overarching goal of this entire scientific endeavor, the meticulous application of selective zinc complexes, the optimization of the intestinal barrier, the reversal of thymic involution, the extinguishing of inflammaging, and the integration of lifestyle and psychological well-being, is the preservation of long-term physiological independence. Independence, in the context of advanced age, is not merely the ability to live alone, it is the absolute freedom from the fear of physical frailty, the capacity to engage robustly with the world, to travel, to interact intimately with grandchildren without the paralyzing terror of catching a life-threatening pathogen, and to maintain the cognitive sharpness required to direct one's own life. For decades, the medical establishment has largely offered the elderly a defensive strategy of isolation and palliative management, encouraging them to retreat from the world to avoid illness. The Selective Zinc protocol represents a radical, offensive paradigm shift, a biologically precise methodology designed to forge an internal armor so resilient that the elderly individual can confidently remain an active, engaged participant in the theater of life. As we continue to refine these protocols, combining the cutting-edge insights of immunosenescence research with the advanced delivery systems of nutritional biochemistry, we are mapping the intricate pathways to a future where the chronological age of 65 is no longer viewed as the beginning of an inevitable biological decline, but rather as a transition into a meticulously managed, highly optimized phase of life characterized by profound immunological resilience and enduring vitality. The intricate biochemistry we have explored, from the evasion of phytates in the intestinal lumen to the precise activation of thymulin in the thymus, and the master regulation of the NF-κB inflammatory pathway, all point to a singular, empowering truth: the aging immune system is not permanently broken, it is simply starved of the highly specific, bioavailable signaling molecules it requires to maintain its structural and functional integrity. By utilizing the precise tools of selective mineral supplementation, guided by a deep understanding of cellular biology and supported by comprehensive lifestyle and psychological interventions, we are unlocking the very mechanisms of biological defense, offering a transformative blueprint for maintaining robust health, unyielding vitality, and absolute physiological independence long into the sunset years of human life.